Assalamu alaikum, dear students. I am Muhammad Rauf, lecturer, Department of Zoology Cast. Welcome to the course Principles in Animal Life 1. This is lecture number 28. The topic is proteins and amino acids. Discussing agenda for the topic will be proteins and their building blocks, amino acids. Lecture outcome. After watching and listening this lecture, students will know about proteins, amino acids, and the importance of their structure to the function they perform. Introduction. A protein is a polymer composed of monomers called amino acids. Amino acids combine with one another through peptide bond in a dehydration reaction and give rise to a protein polymer or a polypeptide chain. In other words, a protein is a chain of amino acids. Proteins include a diversity of structures resulting in a wide range of functions. Proteins are the most diverse biological macromolecules. They are only made up of those 20 different types of amino acids. What gives them tremendous diversity is the three-dimensional structure in which they are molded into. Nearly every function of a living being depends on proteins. Proteins perform a lot of functions in the body, which will be discussed in subsequent lectures. Here we will highlight a few of them. In fact, the importance of proteins is underscored by their name. Greek word proteus means first or primary. Proteins are so much important that even their name itself is an advocate of this fact, which means first or primary. Proteins account for more than 50% of the dry mass of most cells. Proteins are the most abundant organic biological macromolecules in the animal's body. And proteins are instrumental in almost everything organisms do. They have a wide array of functions in the body. Some proteins speed up chemical reactions. Those proteins are known as biological catalysts or enzymes, while others play a role in defense, storage, transport, cellular communication, movement, or structural support. Life would not be possible without enzymes, which are biological catalysts, and most of the enzymes are proteins in nature. Enzymes speed up chemical reactions in living organisms. There are usually multi-step biochemical pathways in which each step is catalyzed by a specific set of enzymes, and without the intervention of those enzymes, those reactions will take a lot of time to complete, and therefore the completion at that particular rate without the interference of enzymes would not be feasible for a living system. Hence, life would not be possible without these enzymes. A human has tens of thousands of different proteins, each with a specific structure and function. Right from the beginning of this course, we are repeating a famous phrase again and again, and we have had a lot of examples related to that phrase too. Here is yet another opportunity to repeat that phrase, which is structure determines the function, and that particular phrase, it holds true for proteins more than anything else, because a protein must be in a proper three-dimensional form in order to perform its function correctly. Proteins, in fact, are the most structurally sophisticated molecules known. Consistent with their diverse function, they vary extensively in structure, each type of protein having a unique three-dimensional shape. So it is the unique structure of a protein which enables it to perform diverse functions. Proteins are all constructed from the same set of 20 amino acids linked in unbranched polymers. When we study carbohydrates, there you will have an example of the branched polymers in which branches emerges from a main stem, like branches emerges from the main stem of a tree. But in proteins, there is no branching, there is only a polypeptide chain involved, and then intramolecular bonding takes place in that polypeptide chain. The bond between amino acids is called a peptide bond. So, a polymer of amino acids is called a polypeptide. Another explanation of this term is that once upon a time the amino acids were called as peptides. So that word fits beautifully in both cases. If you are referring to a polypeptide chain as having many peptide bonds, or you are referring to a polypeptide chain as having many amino acids, so it fits in beautifully in both cases. A protein is a biologically functional molecule made up of one or more polypeptides, each folded and coiled into a specific three-dimensional structure. Now, what is the difference between a protein and a polypeptide? The proteins can be complex or they can be simple. If a protein is composed of a single polypeptide and then this proteins becomes functional, then you can use the polypeptide term and the protein term interchangeably in that particular case. If it is made up of only a single polypeptide, whereas in majority of the cases a protein is usually composed of several subunits, and therefore we cannot refer to the protein in that case as a polypeptide because it becomes a combination of several polypeptides. So in that case we will call it as a protein, like the hemoglobin is made up of 4 polypeptide chains, 2 alpha and 2 beta. So you cannot term that protein a polypeptide because it is made up of 4 polypeptides. Now we will discuss the individual building blocks or the amino acids which combine and form the protein polymers. All amino acids share a common structure. This common structure is shared by all amino acids. This is the basic skeleton of an amino acid, which is shared by all amino acids. An amino acid is an organic molecule with both an amino group and a carboxyl group. Here you can see there is an asymmetric carbon called alpha carbon in the center, and it has got 4 partners. On one side there is amino group, on the other side there is a carboxyl group, and then we have a hydrogen atom, and this particular group is called the R group or the side chain, and it is very important as far as the physical and chemical characteristics of an enzyme is concerned, because it is unique for each type of an amino acid. It gives characteristic properties to an amino acid, and those 20 types of amino acids, they only differ in the composition of that R group. This R group can be as simple as hydrogen present in glycine, or it can be a complicated structure. At the center of the amino acid is an asymmetric carbon called the alpha carbon. The R group, also called the side chain, differs with each amino acid. This is the group which gives characteristic properties to an amino acid. If this group is hydrophilic, the amino acid as a whole will be hydrophilic. If this group is hydrophobic, the amino acid as a whole will be hydrophobic. So it has enormous effect on the characteristics of the amino acid. The R group can be as simple as a hydrogen atom, or it may be a carbon skeleton with various functional groups attached. In the next slide we have an illustration of the 20 different types of amino acids, and recall from the previous slide that the basic skeleton is same for all 20 amino acids, and what make those amino acids different or unique from one another is their R group. You can see here they have shown the R group in the yellow shade, and here in the green shade. This is the R group of those different types of amino acids, and this R group can be as simple as hydrogen in glycine, or it can be a complicated one like present in arginine or lysine. So this R group is responsible for imparting different characteristics to an amino acid. Now here a non-polar side chain is present, and as a result these amino acids are showing hydrophobic properties. Here a polar side chain is present, and as a result those amino acids are exhibiting hydrophilic characteristics, and here they have electrically charged side chains, and they are hydrophilic in nature. These two are acidic, having negative charge, and these three are basic, having positive charge. If you remember, we discussed the histones, and there we discussed that what makes the uh, the histones to be in a strong association with the DNA, it is actually because of the presence of arginine and lysine residues in the histones. Arginine and lysine are positively charged amino acids, DNA is negatively charged. So this electrostatic attraction provides strength to the nucleosome. Here the amino groups and carboxyl groups are all depicted in ionized form, the way they usually exist at the pH found in a cell. So inside the cell the amino acids are present in the ionized form. The amino acids are grouped according to the properties of their side chain. We have already discussed that: hydrophilic, hydrophobic, positively charged, negatively charged, all because of their R group. One group consists of amino acids with non-polar side chains which are hydrophobic. The other consists of polar side chains which are hydrophilic, and acidic amino acids generally have negative in charge due to the presence of carboxyl group, which is usually dissociated at cellular pH. This was the end of the lecture. Thank you very much. You can ask questions directly in the KCMS portal in that particular class activity, or you can also ask questions on WhatsApp groups. Thank you for having me. Allah Hafiz.